Thank you very much and good morning. I feel that it's an honor and a privilege to have been asked to deliver this lecture about something that I'm very passionate about, which is high-speed nature photography. No, that's actually not what I'm going to talk about today, although that talk may be more interesting than what I'm going to talk about today. I'm going to talk about medullary thyroid carcinoma as a model for a disease in which a surgeon can intervene to take out the organ at risk before cancer develops. And this is just the newest accomplishment in a long list of achievements by surgeons over the past 200 years of the modern era of surgery. And it really also represents kind of my life pathway as a surgeon scientist. I have nothing to disclose. In his excellent monograph, Dr. Atul Gawande wrote the following statement: surgery is a profession defined by its authority to cure by means of bodily invasion. The brutality and risks of opening a living person's body have long been apparent, the benefits only slowly and haltingly worked out. The last 200 years have really seen remarkable achievements in the art and the clinical practice of surgery. These include not only the major advancements were the introductions of antisepsis and anesthesia, but also the vascular anastomosis, and then finally opening the chest and operating on a non-beating heart with cardiopulmonary bypass, organ transplantation in humans. And really, just a short time ago, it was the first time that we demonstrated that it's possible to correct a metabolic disorder, specifically the metabolic syndrome and type 2 diabetes, with a surgical procedure, Roux-en-Y gastric bypass or sleeve gastrectomy, which has been shown now in good studies, at least in selected patients, to be the best treatment for these patients, that it exceeds the utility of maximum medical therapy. So again, I'd like to add to it now the ability to prevent cancer from developing. So the address that I want to give today actually sort of summarizes the story of medullary thyroid carcinoma and multiple endocrine neoplasia type 2A as an archetype for a surgical procedure that prevents cancer development. With the background, the short surgical history that I gave you, we can now, it's been achieved today that we can offer an operation to prevent cancer from ever developing or removing the organ at risk when the cancer still remains in situ. And medullary thyroid carcinoma, I would submit to you, and I hope to show you in this talk, represents one of the very first examples of this, and still probably one of the best models for removing an organ to prevent cancer from developing. Dr. Wells, my mentor, who I'll talk about later, defined surgery to prevent cancer as the preemptive operative removal of an organ prior to malignant transformation or while the cancer is still in situ. Now, the essential features for a disease model that lend themselves to this preventative operation include the following. You need to have a hereditary cancer syndrome that involves a very strong genetic predisposition to cancer development. You need to have a diagnostic test that can simply and accurately detect disease allele mutation carriers. The natural history of the disease needs to warrant an invasive surgical intervention without an inordinate risk of morbidity, and for which there's really no better alternative therapy for these patients. You need to be able to remove the organ that's the target organ for cancer development with a high success rate. You need to have the availability of effective medical therapy to replace the function of that organ. And then it's also ideal to have a reliable tumor marker or screening test to detect the recurrence or the persistence of disease. So the story of medullary carcinoma, I think, represents a unique model for a cancer where there has been a stepwise accumulation of findings, and many of these contributions have actually been made by surgeons. And these begin with the recognition that medullary carcinoma actually arises from the very small subpopulation of C cells or parafollicular cells within the thyroid gland that actually derive from the neural crest. And it wasn't really until 1959 that medullary carcinoma was really recognized as a distinct clinical pathologic subtype of thyroid cancer. This was then followed by the findings that demonstrated that calcitonin, the polypeptide hormone calcitonin, is produced by medullary carcinoma, the genetic discoveries that allow us now to perform a genetic test to identify patients at risk, and then this has gone on even farther now to targeted medical therapies. Now, I promised Dr. Potts that I would talk about education a little bit during this talk, and it really fits well, I think, with describing my pathway in the study of medullary carcinoma, because mentorship has actually been very important to me, as I think most of us in this room can attest to. So mentorship really has its origins in ancient Greece as a technique to impart to men, or young men, their important social, spiritual, and personal values. And as we know it today, it's loosely modeled on kind of the apprenticeship or craftsman model, where young people would learn a trade by shadowing the master artisan. Now for me, the master artisan, primarily for my research, was Dr. Samuel Wells, pictured here. And Dr. Wells is an intense individual. He was certainly intense when we trained under him. He had high expectations for us, but I can tell you that he was a fair boss. And whenever you walked into Dr. Wells' office, he made you feel like you could hit a grand slam home run every time you went up to the plate. And of course, those of you who know anything about baseball, it's impossible to hit a grand slam every time you go to the plate. But Dr. Wells made us feel that way when we walked out of his office. Now recently I was asked to address a group of several hundred very ambitious, idealistic, somewhat naive, but incredibly bright undergraduate students at Baylor on the pre-medical day, and to try to explain to them what my personal opinion was of what a pathway in medicine means. And I did this in 2009. I looked back at my slides, and it's interesting to me what I came up with, and especially the last point here. I thought it was important to pursue excellence in academic achievement, to maintain a strong family base and interest outside of medicine, which I'll talk briefly about at the end. And this is really what my mentors taught me, was to be passionate about what we do and be dedicated to caring for the sick. You can see there my final point was to identify key mentors. Now the major influences in my personal development included the role models that are listed here. They start with my father, who is now deceased. My dad graduated from a high school class of about 20 individuals in Dewar, Oklahoma. He was the first one in his family to ever go to college. He put his way all the way through Tulsa University Engineering School, working around the clock at night, and became a successful petroleum engineer in Tulsa, Oklahoma. Dr. William Hillis was my biology professor at Baylor University, and influenced countless young students in their career paths. And then finally, John C. McDonald, 23 years. He was a pioneering transplant surgeon in Louisiana, the chairman of surgery at LSU, and I was lucky enough to marry his middle daughter, who is now my wife, Karen McDonald Lairmore, and he was a major influence in my life. My surgical role models include those individuals listed here that motivated me and that I most wanted to emulate. I think you can recognize the gentleman on the right here. It was really these two guys here that inspired me to go into surgery. Now, pictured here I am in 1988 at the completion of my 4th year in medical school, enjoying a libation with my classmates and mentor. This was at the occasion of the medical school's annual cadaver ball for 4th year students. We had the opportunity to invite a faculty member to sit at our table. And this is who we came up with. Although I don't remember exactly obviously Dr. Potts was imparting some very sage and profound modicum of wisdom to us here as we assembled. I don't remember exactly what the nature of that wisdom was. I think it may have been, as I recall, similar to the advice that Dean Warmer gave Kent Dorfman in Animal House. Yes, okay. I should have started out by saying that parts of this talk are factual and others are possibly lore. But Dr. Potts told me that there would be no questions at the end, so I can basically say anything I want to. ultimately, I overcame the incredible obstacles, both real and imagined, and prevailed, completing my career at Vanderbilt, and I went on, as Dr. Potts said, to go to Washington University. And there, it was a very unique time when I went to Washington University in the history of cloning human disease genes and basically unraveling genetic information. And so why should a surgeon study the genetics of a hereditary cancer syndrome? What business does a clinical surgeon have going into the research laboratory and studying MEN types 1A, 2A, and 2B? Well, it's because all the components of these syndromes are treated surgically, if you think about it. So for MEN2A, the thyroid cancer is the malignant component, and it's treated but with a surgical procedure, a total thyroidectomy. The hyperparathyroidism is addressed surgically. The pheochromocytomas are addressed surgically. So, you know, at the core of this understanding, it's important to point out that cancer is a genetic disease. So the transformation of a cell from its normal phenotype to the malignant one is now known to involve a stepwise accumulation of genetic defects that render the cell unresponsive to normal growth controls. And so the hallmark of cancer is then uncontrolled growth. And these can occur in either oncogenes, which induce activation of stimulatory pathways within the cell, or it can occur in genes that are normally tumor suppressor genes, where their normal function is to impart a brake on cellular growth. There are also genes that involve critical regulatory points in the cell cycle. So if you think about the mechanisms of oncogenesis, there are basically three different types of genes. Oncogenes, which are dominantly active. In other words, if you get a mutation in an oncogene on one of the homologous alleles on the chromosome, it's enough to cause cancer. Tumor suppressor genes involve inactivation of both of the homologous copies of the gene. And then there are also associated DNA mismatch repair genes. Well, this is important because MEN2A is caused by mutation in an oncogene, and MEN type 1 is caused by a mutation in a tumor suppressor gene. So the story of medullary thyroid carcinoma really begins with this cell. It's the C cell of the thyroid gland, shown here with an immunoperoxidase stain for calcitonin. And as I pointed out, these cells actually derive from the neural crest. They're a very small subpopulation of cells within the thyroid gland. And it's really remarkable, at least to me, that medullary thyroid carcinoma was really not recognized as a distinct type of thyroid cancer until 1939, when Dr. Hazard, a pathologist at the Cleveland Clinic, recognized this entity based on its more intermediate behavior. Prior to this time, these more solid types of thyroid cancer had been grouped with the undifferentiated or anaplastic thyroid cancers, but it was clear that there was a subset of them that behaved in a much more moderate biologic fashion. And then it actually was shortly thereafter, in 1961, that an observation that was made by an internist, Dr. John Sipple in Syracuse, was really the beginnings of the recognition of the multiple endocrine neoplasia type 2 syndrome. And so Dr. Sipple was actually not a surgeon, not a geneticist, he was a pulmonologist. And he made a seminal observation while a medical student. And the simple observation that he made, which was inspired by seeing a patient at the time of autopsy, a patient he had taken care of who had both medullary thyroid carcinoma and multiple parathyroid adenomas, was he did a literature search as a medical student, and he noted that within the cases that were reported in the literature, that there was a much higher incidence of pheochromocytoma in patients that had thyroid cancer. And that simple observation really represents the MEN2 syndrome. So just to briefly review, MEN type 2A, which is the most common variant of the MEN2 syndrome, involves medullary thyroid carcinoma, which is the malignant component of the syndrome that develops in essentially 95% of people who inherit the gene. A more variable subset of patients will develop pheochromocytomas, which can be bilateral, and they also develop hyperparathyroidism. And this just demonstrates a section of a thyroid gland that's been removed from a patient with MEN2A, and the critical features here are that the medullary carcinoma is bilateral and multifocal within the thyroid gland, and it corresponds to the middle and upper thirds of the thyroid lobe, which is also where the increased distribution of C cells is. It's very characteristic that medullary carcinoma has a amyloid-like stroma, which is negatively birefringent when you put it under polarized light. And it turns out that it's not amyloid, though. It's actually a polymer of pre-procalcitonin. Pheochromocytomas develop in patients with MEN2, and they can be bilateral. You can see here also that there is a sort of pre-malignant hyperplasia of the adrenal medulla, which is called adrenal medullary hyperplasia, and that precedes the development of discrete pheochromocytomas in patients with MEN2. Patients with MEN2 also develop hyperparathyroidism, and I'm here to tell you, in spite of what you might read in the textbooks, it's usually not multiglandular in patients with MEN2A. It is very much multiglandular in MEN type 1, but it's very common for patients with MEN2 to actually get parathyroid adenomas, a solitary and large gland. This is a demonstration of a patient I took care of early on in my career who had a mediastinal parathyroid adenoma. The MEN2B syndrome is much more rare. It also involves the development of medullary thyroid carcinoma as well as bilateral pheochromocytomas. But these patients also develop a phenotype that's recognizable. They have a very tall, thin, marfanoid habitus, and they develop multiple mucosal neuromas on the tongue and the buccal mucosa. There was a time back in the mid-90s where I think I probably knew about 85% of the patients in the United States that have MEN2B. So I used to travel around with Dr. Wells and screen these people. We would lay them down on their couch on their home in the Appalachian Mountains and put an IV in them and stimulate them with penigastrin. But if you want to make a patient with MEN2B angry, ask them to stick their tongue out, because they've been asked that their entire life. If you look at the the rest of the body, there's actually a diffuse hypertrophy of nerves. This is a split section of the mucosal and ganglionic plexi in the bowel, and they even get hypertrophied corneal nerves on slit lamp exam. Okay, so that's just a brief review of the clinical syndrome. So when I got involved in this as a surgery resident in approximately 1989, this is what was known about the genetics of MEN2. So at this time, this preceded the Human Genome Project, and so only a few genes had been cloned, notably the cystic fibrosis gene, which was one of the very first genes to be cloned. But it turns out that very few human disease genes at that point in time had actually been identified, because most of them either had to have a large chromosomal rearrangement that pointed to where the gene was within this vast expanse of genomic material. It really, few of the disease genes had been detected. Well, Dr. Wells, as a surgeon, had the foresight to assemble a team of expert scientists, because his goal was to identify the mutations that cause MEN2, and he was ultimately successful in doing that with the group that he assembled. I was only a part of that research group, and I can show you some of the other key players. But at that point in time, the genome was not sequenced. So what we knew at that point was that the gene that caused MEN2A had to be located on the long arm of human chromosome 10, somewhere between the gene for interstitial retinol binding protein, RBP3, and the fibronectin beta receptor gene. But this was a very large piece of DNA in the pericentromeric region of chromosome 10. And so the strategy that was used at that time, which is what I spent 3 years in the lab doing, is a strategy that's called positional cloning. And this basically involved starting with a signpost on a marker, sometimes just an arbitrary marker on the chromosome, and then trying to clone up large pieces of DNA with strategies called chromosome walking or chromosome jumping, and then to search within those limited intervals of DNA for candidate gene sequences. And this was a very arduous process. This is the research group at Washington University. It included my mentor, Dr. Wells, and also a PhD geneticist, Dr. Helen Donis Keller. Additional colleagues included Jeff Moley and Jim Howe, who's now at Iowa. And it was really myself and Jim Howe and David Shee who did the majority of the work that I'm going to describe. So this is the strategy that we used. It turns out that Washington University had a genome center at that time with a very, very bright individual named Maynard Olson, who will very likely still possibly win the Nobel Prize. He's been up for it a couple of times. But basically, he designed this cloning system, which is called a YAC, or a yeast artificial chromosome. So I'm going to try to explain this in country boy terms and not lose everyone with the genetics here. But basically, a yeast artificial chromosome, a YAC, is a mechanism to basically propagate very large segments of human DNA. And they're propagated as an extra or additional chromosome in a yeast cell, in Saccharomyces cerevisiae. So all he basically did is put some YAC chromosome telomeres and put them on the ends of a large piece of DNA and created a library that covered the human genome 5 times. Now that's a very simplistic explanation of an incredible resource we had at Washington University, which was a library of YAC clones that covered the human genome about 5 times. So this slide right here summarizes basically about 3 years of work that I did in the laboratory. And basically, at that time, we knew that there were 2 markers, D10S102 and D10S141, and we knew from genetic linkage studies, basically on critical crossover events that occurred, that the gene that caused MEN2 had to be located between those 2 markers. So I spent 3 years pulling out YAC clones from this library and ordering them in a continuous physical contig that basically overlapped this entire region. So you can see here these YAC clones with different markers here that correspond so that I could line them up. And this contig, we called it, it spanned over 1.2 megabases of DNA on human chromosome 10 in 10q11.2. And it continuously covered the entire region between the markers D10S102 and D10S141 that we knew the gene had to be located in. Now, we began searching for gene sequences in there, and it was known that there was a proto-oncogene that was located within this interval called the RET proto-oncogene. And ultimately, again, not solely on my efforts, but those of a large number of people in Dr. Wells and Dr. Donis Keller's research group, we identified mutations in this gene that are associated with MEN2. And this was shown back in about 1992. This just is a representation of the protein. It's a receptor tyrosine kinase that is encoded by the RET proto-oncogene. It consists of an extracellular ligand binding domain, a transmembrane domain, and then an intracellular catalytic domain. And basically, whenever the ligand binds to the outside portion of this receptor, it causes downstream signaling. And so this is a common type of a protein. It's called a receptor tyrosine kinase. And there are many of them now that can be targeted as therapies for cancer treatment. Now, this is actually a fascinating protein. It turns out that 95% of the mutations that cause MEN2A occur within one of 5 highly conserved cysteine residues that are immediately adjacent to the transmembrane domain. And I'll tell you why that's important in a second. Now, it turns out that almost all patients with MEN2B, 98% of them have a single point mutation in codon 918. And there's only one other good example of a genetic disease where a single point mutation it causes almost all of the disease, and that's sickle cell disease. But this is a fascinating protein because it turns out that at least a subset of patients with familial Hirschsprung's disease actually have mutations in other parts of this gene as well. And sometimes Hirschsprung's disease can be associated with MEN2. Now, what this molecule does, actually, when the ligand binds to the extracellular domain, is it actually dimerizes on the cell surface, and that phosphorylates the portion of the cytoplasmic catalytic domain, and that causes downstream signaling. So what actually happens when patients get these mutations in these highly conserved cysteine residues is that the receptor dimerizes without the ligand binding to it. So it's constitutively activated without the ligand binding to it. And this type of overactivation is what actually causes the MEN2 syndrome. So this is really the actual autoradiograph of when the mutation was found at Washington University. I'm enough of a dinosaur to remember the days when we did sequencing on very long polyacrylamide gels and did autoradiography with dangerous substances like P32 and S35. This is almost never done these days. But in the typical pattern GATC here, this mutation causes a codon change of ATG to ACG, and that results in an amino acid substitution of threonine for methionine at codon 918. And because you have both a mutated allele and a normal allele, you can see on the autoradiograph here that there's both a T and a C that show up at this position. So where that little arrow there is, this is the original autoradiograph that identified the MEN2 mutation. Well, why is this important? I started the talk out by saying that surgeons had made many of the contributions for medullary carcinoma, but Dr. Wells's plan to assemble a geneticist and to put a couple of surgery residents in the lab for 3 years to clone the MEN2 gene was not just to figure out what the mutation was. It was because Dr. Wells realized that if we could determine who was affected with this mutation, that we would have the opportunity to take the thyroid gland out in these children prior to the development of medullary carcinoma. So that was his plan from the very start. And genetic testing can be performed. I'm going to kind of get out of some of the thickness of the genetics here, but basically the mutation analysis involves PCR amplification of the important exons, and then they can be cleaved with strategic restriction endonucleases, where they will only be cleaved if the mutation is present. The enzyme won't cut the piece of DNA if the mutation's not there. And this is an example where if you have two bands here, if it's been cleaved, then you have the mutation. You can see in the grandparental level here, the grandfather here is affected with two bands, and all of the SIBs in the second level are affected with two bands. But for all of these children at risk here, it's very straightforward with this DNA testing to tell who's been affected. Is an unaffected patient, affected, 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 unaffected. So back in 1996, we began treating children based on genetic diagnosis alone. And this is an original paper that I published back in 1996 of the first 28 patients who underwent early thyroidectomy. And you can see that some of them had normal stimulated preoperative calcitonin levels. So this was long before they developed clinical, biochemical, or sonographic evidence of medullary carcinoma. And what's really important is that even in the first decade of life, one of these children on final pathology was shown to have cervical lymph node metastases. So we clearly need to intervene early in these patients. The strategy of taking out the thyroid gland as a preventative operation in children with MEN2, Dr. Wells, in typical fashion, reported 10-year follow-up data in the New England Journal of Medicine back in 2005, and he studied these children rigorously. He took 50 patients, children that underwent a preventative operation based on genetic testing. He had 100% follow-up in the patients. And he defined cure as undetectable stimulated calcitonin, which is a very rigorous definition. So even patients who had a detectable but a normal calcitonin level, which indicated they had either normal C cells remaining or persistent disease, were categorized as failures. He only called cure patients that had an undetectable calcitonin level after stimulation. And so just to summarize the results of that study, which basically is the best study still in the literature that tells us how effective it is to take out these children's thyroid gland. The children in this cohort ranged in age from 3 to 19 years old with a mean age of about 10 years. None of them had signs or symptoms of MEN2. They all had a standard operation, a total thyroidectomy, a central level 6 cervical lymphadenectomy. At least in Dr. Wells's hands, we took out all the parathyroid glands and performed heterotopic autotransplantation. That's controversial today, whether that should be done in patients with MEN2. But he studied them 5 to 10 years, and basically the bottom line of the study was that 88% of these children had undetectable stimulated levels up to 10 years postoperatively. So almost 90% of these children are cured long term. There were another 8% who had calcitonin levels that were within the normal range but still detectable on stimulation. So Dr. Wells defined that as a failure, even though it's unclear whether those patients actually still have any persistent disease. And really only 4% of them had clearly evident persistent disease. So it's highly effective to intervene. The only significant factors that Dr. Wells found in his study, that were related to prevention or cure were the age at the time of the thyroidectomy and whether or not the patient had lymph node metastases, which again emphasizes why we need to operate on these patients prior to the onset of cancer development. So this turns out to be the current recommendations for the management of hereditary medullary carcinoma based on genetic testing. The standard operation is to perform a total thyroidectomy and a central node dissection. The management of parathyroid glands is currently controversial. It's critical to avoid permanent hypoparathyroidism when you operate on these young people. And so some feel that it's too aggressive to take out all their parathyroid glands and put them in the arm, although Dr. Wells had excellent results for his immediate fresh transplantation, which warranted that approach. I didn't show you the data, but there were only 4% of the patients in that study that had that still required calcium or vitamin D supplements. So 96% of those children were free of the need for calcium supplements. The current recommendations are to operate on children at approximately age 5 years if they're known to have a genetic mutation. And that's based on data that shows that the earliest lymph node metastases occur in the latter half of the first decade of life. So the earliest reports are in the 6, 7, 8-year range where these children will develop lymph node metastases. So the goal is to intervene prior to that. Now, the patients that have MEN 2B have a much more aggressive behavior of their medullary carcinoma, and we operate on them as soon as the diagnosis is made in the first year of life. The youngest baby that I've operated on for MEN 2B was 8 months old. Now, it can be very challenging to operate on these children. It's a smaller version of the operation we do for adults. And this is actually a very recent preventative thyroidectomy that I did in a 6-year-old child. And you can see here, this is a standard small Wheatlander retractor, so you can see how small the incision is here. That's a Babcock clamp that's on the thyroid lobe. This is the child's head is here and feet are to the left. You can see here the recurrent nerve, which can be identified by this very constant vessel on the perineum here. And then you can see here the inferior parathyroid gland right here, which has been protected. Just for reference, these are 4 silk sutures that are tied here, so that's how small this operative field is. And I think we're always concerned as surgeons when we operate on our patients. You know, I know I was trained that the day that I walk in the operating room and I'm not worried about injuring the recurrent nerve is the day that I need to stop doing the operation. And it makes me nervous every time I do it. But I can assure you that when you walk into the operating room to operate on these children, it's with a great responsibility to not injure their nerve or leave them permanently hypocalcemic. Now, I show this picture with the permission of the parents, and I won't give the names, but I operated on these 2 little girls actually just a couple of weeks ago. And I just included this slide to sort of emphasize some of the components that go into this. These are twin girls. You can only tell them apart by one had no teeth and one had one tooth in the front. So here they are in matching beds at our children's hospital after undergoing their early preventative thyroidectomies. And mom wanted a better picture that was my picture that I took and I got permission to show. But she wanted a better picture of them. So here they are. They're actually doing well. It's very critical. How am I doing on time here? I'm about to wind up here. So it's important to keep in focus that the purpose of this operation is to prevent the child from ever developing cancer. And so it's critical to remove all of the thyroid tissue when you do this operation. You're not helping the child if you're not able to get the whole thyroid gland out. It's also important to perform a very complete central zone lymphadenectomy. Again, in a child here, you can see the trachea, you can see the recurrent nerve that's been preserved here. This is the carotid sheath. And it's important to completely remove all of the lymphatic tissue in the central compartment of the neck. Medullary carcinoma is not amenable to treatment with radioiodine. It doesn't collect radioiodine, so surgical management is the mainstay. So for patients that have sonographic or clinical evidence of lateral lymph node metastases, we also do a very complete functional dissection in the lateral neck. So this is the left side of the neck here. You can see here again the recurrent laryngeal nerve here that's been preserved. The superior parathyroid gland is not as easy to see. It's right there. Here's the internal jugular vein. And again, we've dissected all the levels. Here are the lymphatic components from levels 2, 3, and 4, and level 6, the central compartment, for a patient with MEN2. So there are other examples of hereditary cancer syndromes where the surgeon can intervene. They include hereditary breast and ovarian cancer, but these are, they're not as clean a model as MEN2 is. Patients can have bilateral prophylactic mastectomies. As you know, there are some significant social and other implications to this, cosmetic issues, and also it's not entirely preventative, you know, because it just takes one breast duct cell to turn into an adenocarcinoma. So although it provides great risk reduction, the reduction is not complete, as it is not for MEN2 as well, but it's very high, according to Dr. Wells's data. Another example would be hereditary colon cancer mutations, such as APC mutations in familial adenomatous polyposis, and then the Lynch syndrome. So in summary, early preventative total thyroidectomy in patients with an MEN2-associated RET mutation represents one of the first, and I think perhaps still the purest archetype of a cancer syndrome where an operation is indicated based on a molecular genetic diagnosis with the intent of completely removing the target organ and preventing cancer development prior to its occurrence or when it is still organ confined. And I think, you know, Dr. Halstead is oft quoted with this quote. I won't read it to you, but I think it does kind of emphasize the triumph of the surgeon in taking out the thyroid gland. And this is an elegant operation, and it's something that can now be added to the armamentarium of trying to remove the organ before cancer is developed. And I would be remiss to not recognize Dr. Wells, you know, and his enormous influence on me, you know, professionally, educationally, personally. And I told you that he was he had very high expectations, but he really trained us to work hard and to always strive to improve everything that we did. And he had enormous foresight. The other thing I just wanted to emphasize is a lot of people were having a good time last night and singing and playing music and stuff like that. I've tried to keep that in my life as well, as you heard a little bit of last night. I think it's important to, you know, recreation, it's based on the word recreate yourself, and that's really what you need to do, I think, to continue to be successful. My other hobby is high-speed nature photography, which I'll be going to Trinidad right after this talk to pursue that more. And I really want to, I know Dr. Potts is probably reticent to have me say this, but I want to recognize his influence on me as well. Because when I walked into his office in 1987 as a third-year medical student, I sat down across from him at the desk and I told him I wanted to be a surgeon, and I asked him if he would help me, and he did. And I'm indebted to him. So thank you for inviting me to do this, Dr. Potts.